So the next speaker we have is Dr. Ryan Kelly, and he is here. Just wanted to make sure, is he okay? So I'll do a quick introduction for Dr. Kelly. So Dr. Kelly has been an associate professor in the Department of Chemistry and Biochemistry at Brigham Young University for some time now. And since joining BYU, he spent 13 years as a research scientist at Pacific Northwest National Laboratory. A central theme of Dr. Kelly's research has been the development of new technological solutions for improved biochemical analyses and mass spectrometry-based omics. Recent research efforts have focused on improved sample preparation, separations, ionization, and mass spectrometry for single-cell and near single-cell proteomics. Dr. Kelly has authored and co-authored more than 100 publications and is named inventor on 14 issued and pending patents, several of which have been licensed and commercialized. His work has been recognized with several awards, including 2 R&D 100 awards, the HPLC Faculty Fellowship, and the HTC Innovation Award. So with that, thank you very much for joining us today, Dr. Kelly, and I look forward to your talk. Oh, you're on mute.
There we go. A little
Perfect.
bit.
Okay, thank you.
Okay, sorry about that. Yeah, so we'll be talking about in-depth, high spatial resolution proteome profiling, trying to directly look at kind of develop maps of protein expression across tissues. And a lot of what I'll also be sharing is kind of developed in mammalian cells, but I promise there will be a bit of plant cell data or plant tissue data at the end of the talk. So, so bear with me. Let's see. So most proteomic analyses are performed at the bulk scale. There's just been technological challenges with reducing down toward the single cell level. And so this gives you an average of protein expression across an entire tissue or a biological sample, often averaging across many thousands or millions of cells. So the good thing about that is it gives you plenty of depth of proteome coverage, right? When you're not limited in your sample amount, but clearly information's lost when you're averaging across all these disparate cell types and subtypes. Other approaches towards resolving heterogeneity, one thing we can do, and by the way, each of these approaches is valid in its own right for different applications. So we have different tools for different applications. One thing we can do is kind of look for phenotypically similar cells or regions and pool those together into a single sample that can then be analyzed by mass spectrometry-based proteomics. We can also sort of blindly pixelate a tissue. In our case, we will do laser microdissection, pixel by pixel, isolating tissue with whatever spatial resolution we deem necessary, and then analyze each of those pixels individually to build up a map of protein expression across a tissue. And then finally, kind of looking similar to the pooling method, but taking each one of those cells of interest and placing them in different sample processing containers to monitor protein expression from cell to cell. So these are sort of the different approaches that we use for different applications. And regardless, as we go down in sample input amount, we have challenges in terms of mass spec sensitivity, and we need to do a lot of optimization to be able to achieve an in-depth signal and in-depth proteome coverage from these smaller samples. So we need to optimize, bear with me, turn on my laser pointer, every step of the workflow, from cell isolation, sample preparation, we use liquid chromatography separation, and that needs to be optimized for maximum sensitivity, as does the mass spectrometry analysis and the data analysis. And as we go through these steps, we also need to have an eye towards accessibility to get these techniques out to the community, and also trying to increase our sample measurement throughput using kind of faster liquid chromatography, more efficient utilization of the mass spectrometer, and also multiplexing strategies like our previous speaker described, and what Nikolai Slavov will also be talking about, kind of tandem mass tag multiplexing. And so I'll be focusing on label-free analysis in this talk, but the tandem mass tag approaches such as SCOPE-MS also work well in the nano processing format that I'll be describing. So to begin with, optimizing sample preparation for much smaller samples, we kind of looked at conventional sample processing and using well plates and pipette tips, and we saw the limitations here include we're exposing our samples to a large surface area. Proteins and peptides tend to adsorb to those. We have large volumes, which can reduce your sample or your substrate concentration, leading to inefficient digestion kinetics. And there are often multiple sample transfer and cleanup steps such that these losses can multiply as you transfer from well to well. But there are also some real attractive features of conventional well plate-based processing, in that you can prepare a large number of samples in parallel in a 96 or 384 well plate. These methods are readily automated using liquid handling robots. And because these are open platforms, they're compatible with some of the techniques that we use to isolate and transfer cells or tissues into our reaction vessels, such as fluorescence activated cell sorting or laser microdissection. And so our approach has been to really preserve the well plate and pipetting format, keeping those benefits, but miniaturizing those to a nano well platform to reduce our surface exposure, our volumes, and we've also adopted a one-pot sample preparation workflow to make these possible. Instead of a pipetter, here on the left, you can see we have a robotic nano pipetter that's custom built and can aspirate and dispense below 1 nanoliter volumes. And we keep this in a plastic enclosure that allows us to humidify this to keep from drying out our small samples before we can use them and analyze them. And then instead of a well plate, we have these micro-patterned microscope slides that have these hydrophilic spots that allow us to contain very small volumes. This is work developed with Ying Zhu at PNNL, starting back in about 2016. Ying is now a staff scientist at PNNL and continuing to branch out and develop new technologies related to nanopots, or this technology, which we'll be describing. So we do call this workflow and platform nanopots, which stands for nano droplet processing in one pot for trace samples. And you can see within each one of these little micro patterned nano wells, we're just adding reagents to take our samples through the various steps of proteomic sample preparation, from extraction, alkylation, extraction, reduction, alkylation. Often we'll use a two-step digestion, and then ultimately we'll collect those processed samples. And the only cleanup that we do is a solid phase extraction, and then we're ready to analyze those. So we've really dramatically reduced sample processing volumes, reduced the surface exposure, and presumably enhanced our digestion kinetics as well. So in terms of kind of initial characterization, we just looked at as few as 10 HeLa cells. And what we were able to observe, we were able to identify over 3,000 proteins from these very small samples. And for benchmarking purposes, this showed that we were able to identify more proteins from 10 cells than was previously possible to identify from 500 times larger samples. So samples comprising 5,000 or so cells. So that really did show that it was the limitations in conventional sample processing. And there were also some improvements in chromatography and mass spectrometry that we were putting into place, but it really was the limitations of sample processing that were holding up the ability to analyze smaller samples. So not everybody has a custom-built nanopipetting robot or the ability to make these nano wells. So we have sort of more recently said, well, if we kind of relax our volume requirements just enough to where we can use commercially available liquid handling robots, and if we pre-treat our conventional 384 well plates, would that be possible to still have most of the benefits of nanopots? So that's what we've more recently done. And I should mention that we're sort of splitting what we do in our lab now between nanopots for ultimate sensitivity and autopots for a fully automated sort of push button workflow. So our total volume so we can isolate a sample by kind of limiting dilution or micromanipulation or FACS, or more recently with laser microdissection. And we just put a cover over this well plate that's a kind of a pre-slit cover, and put that onto a temperature cycler that goes into this OpenTron's OT2 liquid handling robot. These only cost $5,000, so it is quite accessible. And then we just do all of our pipetting and our temperature cycling on the robot. So load a sample, pull out prepared peptides, and then place that well plate onto an auto sampler for our LCMS analysis. So as I mentioned, it's not quite as good in terms of ultimate sensitivity as nanopots. We see about a 24% reduction in protein coverage at the single cell level. But then as we go to a little bit larger samples, say 150 cells, that reduction in coverage narrows down to about 12%. So we'll often, for maximum sensitivity, still do nanopot, but for sort of these mesoscale samples, tens or hundreds of cells, autopots, kind of the convenience of autopots becomes preferable. So we can also isolate samples. A non-trivial part of the workflow is getting single cells or small tissue samples into your reaction vessel. So early on, we coupled nanopots with fluorescence activated cell sorting. Because nanopots and autopots are both open platforms, we can just sort cells directly into the wells. And kind of at the single cell level, we were seeing a couple hundred proteins identified from single cells, or upwards of 700 proteins when we're using the match between runs algorithm of MaxQuant or other data processing strategies that are out there. We've also automated the transfer of tissues from, sorry, I think this, well, the little video is not that important, but we can laser microdissect from plant or mammalian tissues and transfer the dissected regions directly into our nanowells for sample processing. And basically the sample preparation workflow doesn't change, just the sample isolation method changes. And we've used this workflow kind of combining laser microdissection with nanopots to map across tissues with whatever spatial resolution you're into. There's kind of a trade-off between proteome coverage and spatial resolution. So in this work, we were looking at some mouse uterine tissue and profiling across there with 100 micron spatial resolution for most of the work, and we could achieve a depth of coverage of about 2,000 proteins per pixel. And you can just see, this is just selecting a dozen or so proteins that we could look at how the protein expression is up or down regulated as we transition from the kind of surrounding stromal tissue into the luminal epithelial tissue of the mouse uterus. And we've done kind of single motor neuron work, microdissecting individual neurons and looking at protein expression in ALS or non-ALS disease. But isn't this conference about the plant cell atlas? Yes. So we have also applied this to plant tissue. You know, where I spent 13 years was Department of Energy National Laboratory. They really don't care about mammalian tissues. They're much more focused on bioenergy relevant systems such as plants. So kind of to prove the technology or prove to ourselves that the nanopots workflow also worked for plant tissues, we just took some readily accessible tomato fruit and microdissected, pulling out kind of the outer epidermal tissue separated from parenchyma or parenchyma tissue, and down to about 8 or 15 cells per isolated tissue region. And so we could look at what's our proteome coverage, what's our and are we able to differentiate the protein expression from these different regions. And you can see here that we could readily, just with a simple PCA analysis, we could readily differentiate the different tissues depending on the region that they were isolated from. More recently, Amir Akami and Ying Zhu at PNNL are applying this general workflow to poplar root and leaf and looking at spatially resolved proteome coverage, in poplar, depending on various growth conditions, drought, salinity, things like that. And they've come up with a very nice protocol kind of optimizing the sample isolation and fixation, embedding, and things like that to maximize proteome coverage from various regions of plant tissue. So I believe that protocol will be out soon, and I think that will be a really good resource for optimizing these nanoproteomic methods for plant tissues. So I haven't had a chance to really discuss more recent analytical improvements to the overall workflow, but there's still a lot of work to do in terms of improving sample processing by miniaturizing separations, using some front-end ion filtering and more sensitive mass spectrometry, more sensitive data processing algorithms, we can improve our single cell proteome coverage from around 200 protein groups. More recently, we're up in the 1,200 to 1,500 protein groups per cell using more sensitive analyses. And so with that, I'll close. Just wanted to recognize the teams that have really made this work possible. Yaron Liang started the plant proteomics at PNNL, and then transferred with me to BYU. Kind of our plant experts at PNNL would be Ying Zhu on the sample processing and analysis side, and Amir and Kim Hickson on the application side of these kind of nanoproteomic approaches. And with that, I'll close, and thank you for your attention, and happy to answer any questions.
Thanks very much for that, Ryan. That's a fantastic talk. Lots of good collaborations and demonstrations of that powerful technology. There's a question here that's just come up. What is the optimal low flow for single cell proteomics?
Yeah, so we have not found the bottom to that question. As you reduce your flow rate for liquid chromatography, you're increasing the ionization efficiency at the electrospray source, and you're also reducing the amount of solvent with associated contaminants that are entering the mass spec per amount of say, single cell protein that you're introducing. And so we have shown pretty good benefits going from a 30 micron column that operates at about 50 nanoliters per minute down to a 20 micron column operating at 20 nanoliters per minute. And then there was some more recent work using open tubular liquid chromatography going down below 1 nanoliter per minute. And the proteome coverage per sample amount is just insane. But we're looking at samples comprising maybe 1/20th of a single mammalian cell or 1/200th, and still able to see some proteins from these really small samples. So yeah, there's room to improve, but a lot of also technical challenges with going below what sort of auto samplers can handle and things like that. So
great. There's one more question here before we move on. Regarding your optimizations that increased your number of proteins, how much of the optimizations are experimental versus computational? For example, I saw you switch from MaxQuant to Proteome Discoverer.
Yeah, so if we kept the MaxQuant throughout that, we would be identifying maybe 900 protein groups, and with Proteome Discoverer, we got to about 1,050 protein groups. And then when we published the paper, researching that data with Proteome Discoverer 2.5 with the Inferis algorithm brings it up to 1,200 or so protein groups. And that's without any MS1 level feature matching, which brings us, up above 1,500 protein groups per cell. So great question.
Fantastic. If there are other questions that come up for Ryan, please post them in the Q&A with Ryan's name tagged to him, and I'm sure he would be glad to answer those. And so with that, I will turn it over to Dr. Wang, who will be introducing the next speaker.